DNA fingerprinting. Data. So today's lab, we're going to be doing a couple of different things. Again, data collection from last week's group projects. So you guys did your group projects last week. You're going to get those plates out. I'm going to get those plates out in the refrigerator here in a little bit. And you do your colony counts off of those. If you have group members that aren't here today, please send that data to them so that they have access to it then as well. The other thing, again, obviously we're doing the DNA fingerprinting lab. I told you we're going to do this crime scene investigation. We have DNA sample from a mock crime scene or crime scene DNA or CS DNA was what it was labeled. And then you have four. We have 5 different suspects, suspect 1 through 5. I don't know why I wrote 4 there. The idea though is we're going to use the DNA samples to tell who the culprit for a particular crime is. But DNA. What are some characteristics of DNA then? So here I have a little short stretch of DNA, but can you guys tell me something about the structure of DNA? Yep, there are nucleotides, base-specific pairing. So DNA, first off, is then double-stranded. One strand of DNA is being held together with a second strand of DNA by base pairing between nucleotides, between specifically the nitrogenous bases of nucleotides. Yeah. Cool. Sorry, I didn't hear you.
Each DNA has a pentose, a phosphate, and a nitrogen.
Yeah, so each DNA is made of nucleotides, and the nucleotide has 3 parts to it. Typically a ribose sugar if we're talking about RNA, or a deoxyribose sugar if we're talking about DNA, phosphate group, and a nitrogenous base. So each individual nucleotide phosphate, some type of ribose sugar, and a nitrogenous base. What are the possible nitrogenous bases then? A, T, C's, and G's, adenine, thymine, guanine, cytosine. Okay, and so for instance, I have them up here. Here's a G, here's a C, here's an A, here's a T. But again, phosphate, deoxyribose sugar, and then the cytosine nitrogenase. This would be one individual nucleotide. DNA is many of these nucleotides joined together into what is called a polymeric molecule. And a polymer just means many, poly, mer means subunits. Many subunits joined together. In this case, DNA is many nucleotide subunits joined together. And then you have one strand of DNA and then a second strand. Again, you have the complementary base pairing. What is the rules for complementary base pairing? What pairs with what? Adenine with thymine, A's with T's, and guanines with cytosines, G's with C's. How do they pair up? What kind of bond holds them together? Hydrogen bonds. And specifically my little red dashes here, these are the hydrogen bonds between, say, a GC versus AT. What do you notice the difference?
There's 3.
Sort of. Yeah. There's 3 hydrogen bonds between C's and G's. And yes, no, they are upside down. We'll talk about that here in a second. We will, no, you're fine on the upside down part. You're right. Your A's and T's are by 2 hydrogen bonds. What does Lexi mean by they're upside down though sometimes?
Anti-parallel.
Anti-parallel. So DNA strands are parallel, but they are anti-parallel. So while one runs with the 3' end at the top and the 5' end at the bottom, the other one is the opposite direction, 5' end at the top and 3' end at the bottom. What holds together an individual strand of DNA? The nucleotides are held together with another nucleotide by phosphodiester bonds, which are strong covalent bonds then. But the phosphodiester bonds form a sugar, phosphate, sugar, phosphate, sugar, phosphate backbone of an individual strand of DNA then. Okay, everybody good? Did I miss anything? Probably, but who cares? Anyways, okay. So again, that's just some of the stuff that we just talked about. Again, is really what we have here that we're talking about. And then also that the 2 strands of DNA form an anti-parallel double helix. So they spin around one another in a spiral shape, but what's called a double spiral or a double helix then. Okay, so suppose you have some DNA samples. You got some DNA samples from suspects and you want to compare it against a crime scene sample. which part of the DNA does allow you to actually compare and contrast from, say, one person to another? The nitrogenous bases, just any single nitrogenous bases, or is it actually a little bit more? Codons and the sequences. So what you're looking at is not an individual nitrogenous base, but the sequence, the order of the A, T, Cs, and Gs throughout the entirety of a DNA molecule. By the way, do you guys know how many nucleotides is in your DNA of a single cell in your body? How long is your DNA chromosome?
No idea.
Huh? You're thinking of 46 chromosomes. Yep, but I'm saying out of all those chromosomes, if you added all the nucleotides together, how many nucleotides would you have in those 46 chromosomes?
Isn't it like billions?
Huh?
Like billions?
Yes,
So it's like
5 billion.
Yeah.
Even something like a bacterial cell, something like an Escherichia coli, has 3.5 million base pairs, nucleotides that makes up its DNA sequences. So we have 5 billion. So there's a lot of DNA in there, but we're looking though is that the sequences of those A's, T's, C's, and G's. So what part of the DNA can you compare and contrast? You can't compare the sugar phosphate backbone. That's the same. It's always sugar phosphate, sugar phosphate, sugar phosphate. There's no variation in there. The part where we see variation is the order, the linear sequence of A, T, C's, and G's in our DNA. So the only variable part of the DNA is the order of the sequence of nitrogenous bases, the order and the sequence of the A, T, C's, and G's. Now, how can we detect sequence differences? Well, we could sequence the entire genome. So we can just look at the sequence of the entirety of one person's genome versus another, but you're comparing 5 billion bases from the first to the last on one person versus 5 billion bases from the first to the last on another person. Anytime you're working in the billions, it's not a good thing. Okay, and so we could do whole genome comparisons. We could compare every base in order. That would be the most accurate, but that is extremely time consuming and extremely expensive. Do you guys know how long it took to sequence the very first human genome, which was accomplished in 2003? So it was not that long ago. Nope, it's done. They can sequence human genomes now.
2 weeks?
2 years.
It was like
10 years,
Oh,
Just like
and it cost a few $billion. Nowadays, though, here's the thing. That entire project was really well funded by the government, but it gave rise to a whole bunch of new technology that did not exist previously. It's just like when they did the space mission, you got a whole bunch of new technologies that are now on things like cell phones and other stuff. So it came out with a whole bunch of things. Because of all the sequencing technology that got invented, put into play, and is now in mass production, you can now sequence a human genome for about $10,000 in about a day. Yes, and the part of the problem was to sequence a human genome for the very first time, it's like putting a puzzle together without the picture.
Oh my God.
Sounds like a fun time.
But now we have, because everybody's genomes is just small modifications here and there. After we sequenced the first genome, we had a map. We had the picture to go off of, and all we had to do was find the differences.
So,
Anyways, so nowadays it's like, but it's still way too expensive and way too time consuming to do that. So that's just not what they do. So it's not necessary to sequence the entire genome. Instead, DNA from a person is isolated and then is treated with what are called restriction enzymes. So we isolate all the DNA from a crime scene sample, or isolate all the DNA from one particular person, and then we treat it with a restriction enzyme. And what a restriction enzyme does, it's a protein, it's an enzyme that breaks apart the sugar phosphate backbone of DNA at specific sequences. Now, what that is, just a fancy way of me saying, is it cuts DNA. You can think of restriction enzymes as little molecular DNA scissors. They come along, they see a DNA molecule, they cut it into pieces. EcoR1, that's one of the enzymes you guys are working today. It stands for E. coli restriction enzyme number 1. And as you can tell, EcoR1 is a restriction enzyme that comes from E. coli bacteria. We've discovered it and we've kind of purified it and we use it for other purposes nowadays, like the lab that we're doing today. But in E. coli, this is an enzyme that cuts DNA. Why would a bacteria want an enzyme that cuts DNA? No, it actually cuts the bacterium's chromosome is very bad and you can kill it. So it's not cutting its own DNA. It's not so that it can mutate. That's a good guess. And actually for cutting or making copies, that might be a good guess too, but it's not what happens. It's actually part of the bacterial immune system. One of the things that bacteria often have to worry about is getting infected by a virus. What viruses do is they inject their genetic material into the bacterial cell and then hijack the bacteria, and they start making the bacteria make copies of viruses instead of copies of itself. Restriction enzymes, if the virus comes along and injects its DNA into the bacterial cell, the restriction enzymes cut up the viral DNA to destroy it so that the virus never reproduces inside of the bacterial cell. So bacteria do have a rudimentary immune system. Almost all organisms have some type of innate antiviral defense mechanisms, for instance. Anyways, EcoR1, though, we purify it and use it to cut DNA sequences. So for instance, EcoR1, it recognizes the sequence GAATTC. And as you can see, that's right here, GAATTC. And what happens is EcoR1 sees that sequence and then it binds to that sequence. And then once it binds onto that DNA sequence, it cuts the DNA molecule. It cuts both strands of it so that it cuts it here and it cuts it there. It does an asymmetric cut. So it doesn't cut it just straight. It cuts it kind of more zigzagged a little bit. But it cuts it into one fragment over here and another fragment over there. So it's cut my DNA molecule from one DNA molecule into two.
Know how much to cut.
It binds to that sequence and that triggers its cutting. And then once it's cut, it comes off of there. So it'll only cut at that specific sequence though. So where do restriction enzymes come in to the activity we're doing today? What we're doing is we're comparing DNA using those restriction enzymes. We isolate DNA from suspects and from the crime scene sample. We treat them with the restriction enzymes. But if there are differences in DNA sequences, and there are differences in your DNA versus another person's DNA, sometimes it's just a point mutation here, like a single base pair, like a T changed to an A. Sometimes actually we have what are called chromosomal inversions. Sometimes parts of our chromosomes can just flip around. Instead of being in this direction, they flip around and they're on this direction. So it turns out all the time we get these chromosomal inversions where part of the chromosome just flips around. It's real weird, but it happens fairly common. It doesn't change any of your genes, but it changes the order of the genes and creates some differences that can be detected in your DNA sequences. So these differences in the DNA sequences, what they do is they change where some of these restriction sites are at. So EcoR1 might bind here, here, here in one person, but now EcoR1 in another person might bind here and here only. And maybe there's this point mutation in this other site that destroyed it. What that means is you get differences in where the EcoR1 or other restriction enzymes bind. The DNA will then cut differently into different patterns. And then you can generate DNA fragments with differing lengths that will then be used to map one person's genetic material from another, from another, from another. But you can see this is, for instance, DNA samples from person A, person B, person C, person D. And this generates what's called a restriction fragment length polymorphism map, RFLP maps. And this is what you probably have seen on the TV shows and the CSI when they pop them up. What they do is just isolate DNA from somebody, they cut it with restriction enzymes, a cocktail of different restriction enzymes. They get these RFLP maps, restriction fragment length polymorphisms. Again, they're just called restriction fragments because you generate them with a restriction enzyme. They're different lengths, and there are many, many different shapes and lengths that result out of this. but every person is going to get their own unique RFLP map. This is basically their own little DNA molecular fingerprint. You guys have probably heard how everybody has different fingerprints. And once you do this technique, you get your own DNA fingerprint, hence the name of the lab today. Questions about that then? Okay, so what you guys are doing, I had you set up those samples just a second ago. You added your enzyme mix. It was a mixture of EcoR1 and PST1 restriction enzymes. We put it in with your DNA samples. Now they're sitting at 37 degrees Celsius. And what's happening is those enzymes are binding to the DNA and cutting it into fragments. But they're only cutting it at their special recognition sequences. Again, EcoR1 will bind on this GAATTC sequence and only cuts at that spot. Okay. And so you get some different RFLP maps. Now, it's not going to be a big, complicated one like this one. It's going to be probably only about 4 or 5 bands. But we're just doing a small scale version of this, but it's really what's done in practice then as well. Okay. So again, we're going to be using these mock DNA samples, just a shortened version of an individual genome to just make for faster, easier evaluation. But in practice, it's not that different if you're actually doing it on a real crime scene samples. You digest your suspect DNA, crime scene DNA, treat with friction enzymes, and attempt to detect differences in the DNA sequence and the links of the DNA molecules produced. With different DNA sequences, you get different restriction fragments, and you can visualize those differences and compare suspect DNA to the crime scene sample. All right, so, right, we already did that a little bit. So I 77, blah, blah, blah. I just did it in a little bit of different order today. Okay. So we still have about 10 minutes or so till they're ready. So I'm just going to keep on going here and talk about what we're going to be doing for our next steps. Restriction digestion of DNA creates, again, DNA of different lengths, sizes. And then you can actually generate a map based upon those different lengths. But the question is, how do you actually even make this map in the first place? Right now, you're just going to have a bunch of DNA mixed together in a tube of different sizes. It's all floating around in the liquid. How do you separate out the DNA, all of those different sizes, into more of a map? And how do you then visualize those DNA fragments? You guys might have noticed, did you see any DNA floating around in tubes when you were working with it a second ago? Yeah. DNA is microscopic. We can't see it, so we need a way to visualize it as well. Well, this is where a technique called gel electrophoresis comes into play. And with gel electrophoresis, what we're just going to talk about is purpose of it here real quick. How does it work? And how can it allow you to visualize DNA? And then we're going to do it. Gel electrophoresis, what's its purpose? It's a technique to separate nucleic acids or proteins based upon the size of the DNA, RNA, or protein molecule. So it separates DNA or RNA or protein based upon the size of the molecule. How does it work? It uses an electrical current to force, to push and pull proteins or DNA or RNA through a gel matrix. And it kind of is almost a little bit like Jello when I say gel. And then because of the way this works, then the molecules, the DNA, for instance, gets sorted into bands based upon their size. So they move at different rates based upon their size. Okay, so focus. What we're going to do is literally today, I'm not going to really talk about too much about protein electrophoresis because that's a little bit different. We're going to focus more on DNA and RNA electrophoresis. Again, we load DNA and RNA into a gel, and then we apply an electrical current to it. Why can we use, and that electrical current is then going to apply force. It's going to push and pull the DNA or RNA through the gel. Why would an electrical current, though, affect DNA and RNA and cause it, put a force on it, cause it to move through a gel?
The phosphate group has a negative charge on it.
Yeah. Remember, there's a lot of phosphate in DNA. There's a phosphate, sugar, phosphate, sugar, phosphate, sugar backbone. Every phosphate has what charge? Negative. And that means DNA and RNA are both highly negatively charged. And the negatively charged things will interact with electrical fields. So we can apply an electrical current, apply an electrical field, and that will interact with the DNA molecules because they're highly negatively charged. So what it has, again, all DNA molecules are negatively charged. All of them are roughly the same shape. So what we can do is we have our gel. So this is what our gel is be, and you guys are going to practice with the gels here in a second. There's some wells in it. So it's kind of hard to see, but do you guys see how there's these little holes or wells in it? You're going to load your sample into the well. And then how you guys are going to do your crime scene sample, suspect 1, suspect 2, suspect 3, suspect 4, suspect 5. You're going to put them all into the gel. They're going to sit down in this well, but then we're going to apply consistent electrical field. Not just that, though, we're going to apply a negative charge at the top of the gel next to the wells, and we're going to apply a positive charge at the bottom of the gel. Well, what does negative charge do to other negatively charged things? They repel it. And what does a positive charge do to negatively charged things? Attracts. And remember, DNA is negatively charged. So the negative charge is going to repel the negatively charged DNA downwards towards the bottom of the gel. And it's going to get attracted to this positive charge again at the bottom of the gel. So that's going to be, again, that's why I'm saying it's pushing and pulling the DNA. You guys might have noticed I used that language earlier. The negative charge pushes, the positive charge pulls, attracts. Does that make sense, everybody?
So I see on the figure, it says the longer molecules are towards the negative charge.
Yep.
Wouldn't it be towards the positive charge though, since there's more negative?
That is actually a great question. And we're going to cover that here in a second. So DNA, and that is right. The longer molecules, DNA with more nucleotides are longer, the bigger ones. Those are actually going to have more trouble moving through the gel, though. Why are they going to have more trouble moving through the gel? Does anybody know? Lexi? Okay. Sorry. I'll use my analogy here. Say you have a forest of trees. Trees are pretty thick. You know, it's a very crowded forest. What's going to move easier through the forest, a mouse or an elephant? A mouse. When I say a gel, I literally mean a gel matrix. When you make a gel, you mix some stuff in the water, you heat it, you let it cool down, and it cools down, but it forms literally almost like this net-like array that holds the gel together into this, and it holds the gel together in kind of a semi-solid. But the problem is if you have a big, long DNA molecule, it's got to basically move around through this gel polymeric network. And if you have a longer molecule, it's more likely to bump into the gel matrix. In essence, the longer molecules hit the gel matrix more often, that slows them down. If you have a big elephant moving through a forest, it has to really, sometimes it can't fit between trees and have to kind of take a more circuitous route to get through the forest. That slows it down a lot. Whereas something like a mouse moving through a forest, it's just like zoom, 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 zoom. It doesn't really have to deviate from its path hardly at all. Bigger DNA molecules generate more friction against the gel, and that slows them down. So they have slower movement. Smaller DNA molecules generate less friction against the gel. They just bump into it less often, and therefore they move more quickly. So yeah, wherever you loaded your sample on, the closer it is to the wells that you loaded it, the bigger the DNA molecule. They don't move very far. Small DNA molecules are going to move much further down the gel. So longer molecules towards the wells, shorter molecules away from the wells. Questions about that? All right. So we've loaded our DNA onto the gels. We ran the gels. We still can't see the DNA. DNA is tiny, microscopic. So we can't see molecules in a gel once we're done separating them unless we do a special staining procedure. So we need to stain the DNA molecules to get them visualized. You might notice my DNA molecules here are glowing under UV light. So what's happening is this gel, the reason it's kind of purplish is because they're hitting it with UV light. And that causes, and then they're glowing, they're fluorescing, which then allows you to see them. Why are my DNA molecules fluorescing under UV light? Well, no, UV light does not normally do that to DNA molecules. We have to apply a special stain to it. So for DNA and RNA, you stain DNA with a chemical called ethidium bromide. Ethidium bromide, or ETBR for short. Fluoresces a purple violet color under UV light. Ethidium bromide binds the DNA, but also fluoresces under UV light, and that allows us to visualize any DNA molecules that are in the gel. Okay, that's going to allow us to see them. Questions about that? So the ethidium bromide is already in the gels. So it's already in there, waiting for the DNA to bind onto it. Here's the important thing though. Always, 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 always wear gloves when working with the gel electrophoresis equipment. Any of it. Ethidium bromide is great at visualizing DNA. It's a stain that binds DNA, fluoresces under UV light, allows us to visualize the DNA. Problem is, ethidium bromide is also a carcinogen. It's a chemical that can act as a mutagen. It causes mutations in the DNA, and mutations in the DNA can then lead to cancer, hence it being a carcinogen. Again, ethidium bromide binds to DNA. But when it binds to the DNA, it bends the DNA molecule just a little bit, and then it can actually cause mutations in the DNA molecules. Again, mutations doesn't mean you're going to get cancer. It just means you're going to get at a higher risk of cancer. You guys know what cancer really is? No, it's a mutated version of a normal cell that has unchecked cell division. Just keeps dividing, and then forms a mass called a tumor. It's not one single mutation does not lead to cancer. It's usually a combination of 5 to 12 mutations in just the wrong set of genes that leads to a cancerous cell. So, but it's a mutant version that has unchecked cell division. And that's what most cancers are, for instance. But yeah, mutations, the higher the rate of mutation, if you get mutations in cells, it leads to higher risk of cancer. Questions about that? Okay, when the gel is done running, then we're going to use UV light to look at the gels and interpret them. And I'll take you across the hall to our machine to do that. And then we'll basically look at those as an RFLP map of the crime scene DNA sample, compare them against the crime scene DNA sample and all the suspects, and figure out which one is the suspect that then did the crime. One thing about that, does it necessarily mean that if a suspect's DNA matches the crime scene DNA, does it necessarily mean for sure they did the crime? Well, it kind of depends a little bit. Actually, even with siblings or parents, you're going to generate a different RFLP map. You only share half your DNA with your mom or dad and with a sibling. So no, it just basically means, it depends on where you get the DNA sample from. So yeah, if there's a bunch of blood at the crime scene, and that's where you get your suspect, you get your crime scene DNA from. Yeah, if a suspect was bleeding all over a crime scene, that's probably not the best sign. But it really just places them at the crime scene at some point. Does that make sense, everybody? Yeah. Also, do you guys know how they compare fingerprints? Yeah, they look at the loops. How do you think they actually do that, though?
I don't know, in the TV shows, they just like cross-reference them.
The computer does it, right?
Yeah.
Yeah, that doesn't happen.
Aw.
There's no software for interpreting fingerprints. It's too complicated. There's no AI algorithms for that type of thing. You know what actually happens?
Don't they put like lead or something on your finger and just put it on?
No, yeah, but then somebody has to look at the fingerprints. And literally, somebody who's trained to look at fingerprints just looks at the fingerprints and says, I think that matches. Yeah, ruining all your TV shows. Yeah, actually, if you give them any extra information, it's shown that they tend to have confirmation bias. They tend to say there's matches where there aren't matches. If you give them any extraneous information other than here's of 2 fingerprints, do they match or not? If you say, here's this from a suspect who eyewitnesses also placed at the scene, they're more likely to say that fingerprint matched even when it doesn't actually match. Even with a trained person to look at these. So yeah, anyways. No, okay. Okay, That's my lab lecture, but we're going to talk about a few other things. But we're going to get started with the gels first. A couple quick things. Assignment. There is going to be a pre-lab worksheet due after Thanksgiving break. So pre-lab worksheet number 10. This is going to be over the mitosis meiosis lab. There was one due today if you guys didn't get it done as well. Pre-lab worksheet number 9 was due today. So if you didn't get that in, do that one. So we will have a pre-lab worksheet on November 30th due. Over the mitosis meiosis lab, we're going to do them next week. How many of you are going to Bio 1 right now? Where are you guys at in lecture?
We just took our fourth exam yesterday.
So you haven't even started mitosis yet.
No.
Okay, I'll figure that out. Sounds like you're a little behind you. Normal, but
We definitely are.
it happens
We
some
are.
days. UV yeast paper is due Friday, December 3rd by 5 p.m. I sent out the assignment description for that. You guys are collecting the last bit of data that goes into that paper today. We're going to get our gels started here in a second, but then I'm going to talk a little bit more specific. After we get those gels up and running, it's going to be doing your colony counts while the gels are running, but it's also going to be, I'm going to spend some time just to go through the paper real quick with you guys and answer any questions you might have about the paper. But first, we're going to go ahead and set up our gels. Okay, so paper. That was something I did want to talk real quick while we're waiting for those gels. So now we got the gels running. I'm going to talk about the paper while those gels are running because we have a bit of time. Paper. First off, usually, again, it's always best to start off with the methods and the results section. Again, that's always the two sections I recommend to you guys that you write first. With that then, though, thinking about the method section, again, the method section is going to include the parts of the project that you did, and there's 3 main parts of the project. The percent survival portion of the experiment, the percent mutation portion of the experiment, and then your group project that you guys designed, carried out, and are collecting data for right now. Okay, so those are all 3 things you got to describe in the method section. Again, the method section should be in past tense. You're describing stuff you already did. It should be detailed but concise. So the method section should be very what's called information dense. You should try to say as much as possible in as few words as possible with that. But everything that you describe should be that somebody should be able to repeat it. Okay. So that's kind of the goal with the method section. And most of you did pretty well on the method section of the papers. By the way, the papers, I know I'm a little late with grading those, but most people have their papers. I got like 2 or 3 more to grade, and I'll have those done tonight. But most people actually have their papers now. The grade is posted to Brightspace, and you can access the feedback through the Ensign paper Dropbox. So also take a look at your previous papers to see if there's anything you need to improve upon for the method section. But most people did pretty good with the method section. Results section is a mixture of a textual portion and then the figure graph portion. How many graphs do you think you guys need for this particular paper? 3. What should be in each graph?
Percent survival, percent mutation.
Yes, so yeah, the 3 graphs should be the time of UV exposure versus percent survival for one of the graphs. Time of UV exposure versus percent mutation for the other graph. And then the final graph is going to be over your group project. And if you guys did your group projects, I think most of you just looked at the ability to kill. You didn't look at mutation rates, but that's still going to be percent survival for your different fabrics versus your, so in that case, it's a lot of times, for instance, if you did fabrics, it's percent survival on the y-axis, the fabrics you used and the control on the x-axis. You did sunscreen, it's going to be your sunscreens you use on the x-axis and your control on the x-axis. And again, percent survival on the y-axis. So that's what you're going to be doing for figures. Is there anything else? What else should the figures have?
Title and a small like or sentence.
The figure number, figure title, and a figure caption should be beneath each figure. Anything else? Error bars should be on all of your figures. So I sent out the class data set. From that class data set, you should be able to calculate standard error like you did on statistics assignment number 2, which I got graded and how you guys have feedback on. And then you should be able to add the error bars to all 3 figures then as well. The results section, probably also a good idea to have 3 different paragraphs, 1 for each figure. For each paragraph, start with a purpose statement, what you want to define, and then describe the results in detail and any trends or patterns observed in the results. And make sure you use parenthetical figure citations too, like this came from figure 1. Questions about the methods of results then? Couple other quick things then. Intro, discussion. I don't have too much to tell for you guys. But remember, in the intro, what you're focusing in is the concept that's being explored in this paper. And what's the main concept that's being explored in this paper? Effective what causing gene mutation? UV light, UV radiation. So probably in your intro, it's a good idea to talk about, I don't know, ultraviolet light. Then its impact, and what you're doing is an experiment to basically determine how it does impact both the survival of cells and the mutation of cells. Okay, so you really do need to have a good bit of background on UV light itself in that scenario, though. Again, with the intro should generally be, there's several different ways you can take it that are all correct. But you need to basically be starting off very broad, very general, and gradually get more specific to your actual experiment itself. The reason you start broad is the reader needs some context. They need to understand a few things before they're ready to know about your experiment. They also need to understand that context to know why your experiment is important. So you have to set it up. You do not go straight into, we did an experiment in the intro. Okay. You go start off with broad general background information that is necessary context for a reader to then understand the scope and the importance of your experiment. Should you be using citations in the intro? Yes. And probably quite a few. Any fact, data, figure, any knowledge that is not, anything that's not common knowledge, you need to cite a source for where that came from. I like primary or secondary articles. You guys remember what a primary versus a secondary article is? What's a primary article then? So it's a researcher did an experiment, the researcher then wrote the article and they published it in a peer-reviewed journal. So it's a first-hand account of research, a written account of a research experiment written by the researchers that did the experiment. What's a secondary article then? It's what's called a review article. The better definition of what a review article is it summarizes the work from several different primary articles in one spot. So a secondary article is basically, here's all the stuff from primary articles and we're summarizing it all. And it's usually still written by an expert in the field. What kind of source is your textbook or your lab manual? Tertiary. Okay, one thing to keep in mind is you guys are required to have at least 2 total primary and/or secondary references for this paper. So you have to have at least 2 total primary or secondary. There can be any combination. It can be 2 secondaries, 1 primary, 1 secondary, 2 primaries. But you need at least 2 primary or secondary articles as part of the citation requirements for this paper. You can still cite the lab manual or the textbook. Those are fine to use, but they're tertiary sources, so they don't count towards that requirement. Why am I emphasizing that? Again, I want you guys to be able to search through scientific literature and find good sources of information, not random stuff on the internet that then leads to conspiracy theories and stuff like that. Anyways. Actually, one of my projects in my microbiology course is microbial mythbusters. Take a common kind of myth about microbiology, and yes, it could be like vaccine efficacy or vaccine side effects or a bunch of other stuff, but then basically go through where that myth came from and what's the actual data that's published in professional sources show instead. Why is it a myth, basically? So that's, again, I want you guys to be able to sort through good versus bad sources of information, because there's a lot of bad sources of information out there. And sooner or later, you come across one and kind of fall for it. Even me. Yeah, my wife asked me to research something that her dentist recommended, and I can't find the right answer. She's very disappointed in me. It's like this $800 perio tray thing. Anyways, she's like, does this actually work or not? And I'm like, I don't know. I can't figure it out. Anyways. I spent like a solid 2 hours searching literature sources and couldn't find any professional ones. But Lawson did just recommend it despite that. I don't know. Anyways, I was very disappointed in myself. All right. So questions then about the intro section. Remember, also the final paragraph of the intro section should be purpose. That's where you really get into the experiment. Purpose, hypothesis, methods to assess the hypothesis in that order. Discussion section. Probably 4 paragraphs for your discussion section. 1 paragraph on the survival portion, 1 on the mutation portion, 1 on your project portion, and 1 summary paragraph at the end. In each one of those first 3 paragraphs, so again, it's a good idea to restate your purpose, briefly summarize the methods, briefly. But then any trends or patterns you provide rationales, possible explanations for why you saw what you saw. So explanations for those trends, that's your conclusions. That's your discussion section. But what you're also doing then is comparing what you found against other similar research that has been done. So the other big part of the discussion section is citing outside sources and comparing and contrasting your findings against other research. Questions about that? Final summary paragraph just summarizes all the main conclusions, but also big picture. Why is it all important? What's the applications for this? One of the things that might be that you guys haven't thought of, why would we want to see how well sunscreens work or how well fabrics work? Well, we can use yeast as a stand-in for other types of organisms. Anyways. Lit cited abstract summary of the intro, methods, results, conclusions, in 250 words or less, but I really want 150 words minimum with a 250 words max. Title is not UV use paper. Title is a descriptive full sentence of your experiment. And the title is actually harder this time than the previous time. Because you had both the percent mutation, but also the protection from UV light portion. Think about how you can do that title. It's actually a harder title to make this time around. I'm not saying it's an impossible one. It just takes a little bit of thinking about it. I get a lot of, I tend to get more awkward titles for this particular paper than the first one. All right, questions then about the paper, anything? Friday, December 3rd by 5 p.m. I'm a little loose with the day. I would probably like by December, if you want any feedback on the paper, if you want me to read through drafts of the paper or individual sections of the paper, it's best to get them to me by December 1st, which isn't that far away. Also, again, for most of you, I've already got your papers graded and the comments are back. There's a couple more I got to do this evening. All right, otherwise, finish counting. And then we'll take a look at the gels here in a little bit.